Welcome to this explainer. Today, we're taking a microscopic architectural tour inside your body's incredibly organized immune fortress, the lymph node. You know, when you really look at the structural biology of how our bodies keep us safe, the precision is it's absolutely fascinating. We're going to explore the distinct layers that make up these tiny biological filters and see exactly how they identify and fight off infections. Let's get right into it.
Here's our roadmap for today's explainer. First up, we'll meet the lymphatic system. Second, we'll go inside the lymph node. Third, the outer cortex. Fourth, the inner cortex. And finally, we'll hit the medulla.
All right, Section 1. Meet the lymphatic system, an essential immune network. So, as a quick refresher, the actual fluid being filtered here is called lymph. Basically, it's interstitial fluid that's drained from your peripheral tissues into this vast network of lymphatic vessels. The ultimate goal of these vessels is to return this fluid back into your bloodstream. But before it gets there, it has to be checked for threats, right? And that is exactly where our lymphoid tissues and organs come into play. They're heavily packed with lymphocytes and other white blood cells, literally standing guard.
Now, what's really interesting here is how we separate where these immune cells are made from where they actually go to work. We've got our primary lymphoid organs, that's the thymus and the bone marrow. You can kind of think of those as the training and manufacturing centers for lymphocytes. Then we have the secondary lymphoid organs. This includes the tonsils, the spleen, the mucosa-associated lymphoid tissue, or MALT for short, and of course the lymph nodes. These secondary organs are the active deployment zones. It's where the immune system directly interacts with the lymph to mount a defense.
Moving right along to Section 2, inside the lymph node, the encapsulated being.
To really grasp the scale here, these biological water treatment plants are incredibly tiny. I mean, they usually measure just 1 centimeter across their short axis and maybe up to 2.5 centimeters along their long axis. But despite their small size, they are densely packed and they're perfectly encapsulated in connective tissue. Their entire purpose is to support your immune system by continuously filtering the lymph to identify and fight infections. So yeah, they're small, but they are incredibly mighty.
Okay, Section 3, outer cortex, the B-cell hub located just beneath the capsule.
This outer zone is just absolutely bustling with targeted activity. When you look at it, it's structured into these really distinct spherical nodules or follicles, completely packed with B cells. And right in the middle of these follicles, you'll find what are called germinal centers. Actually, much like the follicles you'd find in the spleen, these germinal centers are active transformation zones. It's exactly here that B cells actively differentiate into plasma cells, basically gearing up to produce the antibodies your body needs to fight off identified invaders.
Now, as we move deeper, there is a really stark microscopic contrast between our layers. On the left, we've got the outer cortex with those distinct, clumpy B-cell hubs we just talked about. But on the right, as we step into the inner cortex, the landscape completely changes. It loses those nodules and becomes much more homogeneous. Microscopically, it takes on a more eosinophilic or pinkish appearance. And functionally, well, it shifts from being a B-cell domain to being populated mostly by T cells.
Which brings us to Section 4, inner cortex, the T-cell zone, also known as the paracortex.
Aside from the massive presence of T cells, there's a really fascinating structural oddity that lives in this inner layer. They're called high endothelial venules, or HEVs for short. These are highly specific blood vessels that are entirely unique to the inner cortex. In fact, if you're looking under a microscope, spotting these HEVs is exactly how you can differentiate the inner cortex from the deeper medulla layer. And they serve a highly specialized, absolutely vital purpose for your immune system.
So the crucial point here is understanding why they look different. In typical venules, the endothelial cells that line the vessel walls are quite flat. But in high endothelial venules, these cells are unusually tall, which is obviously where they get the name high. And this isn't just a random architectural choice, right? Form perfectly follows function here. These uniquely tall cells actually allow lymphocytes traveling in the blood to efficiently grab on, squeeze through, and physically exit the bloodstream to step right into the surrounding lymphoid tissue exactly where they're needed. It's brilliant.
All right, Section 5, medulla, the final filter, the innermost region.
If you're ever looking at a histology slide, you can instantly spot the medulla. Why? Because it looks much lighter and brighter than the cortex above it. And it's not a change in cell color, but rather a change in cellular density. The medulla simply isn't as densely packed with cells. Instead, it's full of open spaces, or areas that form what we call medullary sinuses. Think of this as the grand collection zone, where all the lymph that is just filtered through the cortex pools together before it finally exits the lymph node.
Surrounding and shaping those open sinus spaces are irregular structures known as medullary cords. And these cords house a very specific cellular cast of characters. Packed inside, you'll find macrophages, B cells, and plasma cells, alongside a few blood vessels. These immune cells are positioned perfectly along the cords, basically waiting to interact with the filtering lymph one last time. They're making sure absolutely no pathogen slips through the cracks before the fluid moves on.
So let's pull this all together and map out the actual plumbing system of the lymph node, tracking a single drop of lymph as it flows through. First, lymph arrives from afferent vessels and enters the subcapsular sinuses, located just underneath that outer connective tissue capsule. From there, it follows the capsule's extensions, which are called trabecula, flowing deeper into the cortex through the trabecular sinuses. After percolating past all those B cells and T cells, it eventually collects in the innermost medullary sinuses. And finally, having been thoroughly cleaned and inspected, the lymph exits the node entirely through the efferent lymphatic vessels. The whole shebang is a perfectly orchestrated flow.
As we wrap up this explainer, I really want to leave you with this thought. We've just spent all this time analyzing the microscopic architecture of a single 1-centimeter structure. But right now, literally as you listen to this continuous, perfectly organized flow of fluid and immune cells is actively happening in hundreds of these tiny encapsulated biological filters scattered all across your body. The sheer scale and ongoing hidden efficiency of your own immune system is nothing short of miraculous. I hope this structured journey inside the lymph node has given you a deeper appreciation for the complex world operating just beneath your skin. Keep learning, keep exploring, and until next time, stay curious.